Today is February 3rd, 2021. Today's title is Mitosis. We've got the same objective from yesterday. So this is the second day of our third unit. Same objective from yesterday, but this time we're going to hone in specifically on mitosis. So we're still thinking about the cell cycle as a whole, but we're going to hone in specifically on really the second part of the cell cycle, which is mitosis. The essential question that we will seek to answer by the end of class: how does a cell pass its genetic information to the next generation? Can anybody hear me?
I can hear you.
Okay.
Yeah.
So let's see. Abigail, you may just need to leave and come back for a second. If that doesn't work, then I'll try something else. But just leave and see if it'll work when you come back. And you can also see if it works if you turn on the captions as well. There's an option to use captions. All right. So we want to be in the habit of taking strong notes, effective and efficient notes. That often starts right now with writing down some important information from the introductory slide. Those of you who have visited our YouTube page will notice that this introductory slide is the cover art for all of the YouTube videos. So it tells you exactly what's going to be covered in that lesson. It's important that you all know, and you can come back to your notes and check it out when you're going through your study process. So write down some information here over the next 5 seconds or so as I pull up the YouTube link for us. For those of you who aren't aware, there is a YouTube playlist with all of our recorded lessons. So far, I'm up to 12 views over 18 videos. So not too many of you are tuning in. That's okay if you are attending class regularly, but that option is there for you to use. I will also, as we get closer to the midterm, be updating. Thank you, Sally. I will, as we get closer to the midterm, be updating some review videos, uploading some review videos for you as well. That'll be shorter and just give you kind of like the bang for your buck, the meat of each standard. So be on the lookout for that. All right, so let's review yesterday's lesson, which was about the cell cycle. There were really 3 phases of the cell cycle. And the first phase is really the longest, and it's broken down into 3 separate steps. Does anybody remember what that phase is called? The longest phase of the cell cycle?
The end phase?
Interphase, yeah, good. And what's the first step of interphase? G1, excellent, Sally, thank you. Come over here. And the G could stand for gap. It can also stand for growth. But in this first G1 step, what's happening is the cell is just growing. And it's also doing whatever its job is. So every cell, of course, has the ultimate goal of producing some type of protein, whether it be a hormone or an enzyme or several different proteins. So in this time, the cell is performing its job and it's also growing. What comes after G1? Potentially the most important part of the cell cycle.
Synthesis.
Synthesis, wonderful. And what happens during synthesis?
The DNA replicates.
Wonderful, thank you, JT. So we've got to replicate the DNA before we go through a process of cell division. There could be serious consequences if we do not replicate the DNA first. So synthesis is extremely important. What comes after synthesis?
G2.
G2. Thank you, Melissa, and someone else who just spoke. I think it was Dijon. G2. And again, here the cell is growing. It could stand for gap or growth. The cell is continuing to grow, but it's also increasing the number of organelles it has. So we want more mitochondria, more chloroplasts, more vacuoles, more ribosomes. And the reason for this is that the cell is getting ready to divide. It's preparing itself for cell division. These three steps are of course, known as interphase. And we thank Lance for giving us that term. It is the longest phase of the cell cycle, is where most cells spend the majority of their lives. But then after this, most cells will also reproduce, and that's called M phase or mitosis. Does anybody know what comes last? What's the last phase of the cell cycle? It's a weird word.
Cytokinesis.
Thank you, Abigail, and thank you, Sally. Cytokinesis. Good. So this is a simple depiction of the cell cycle. It might make sense for you to try to draw something like this in your notes. I am a big fan of graphic organizers. And so having some type of pie chart like this that shows you that it's an actual cycle, shows you the different parts, that can be helpful. We're going to build on this M phase portion today. We'll talk about what exactly happens in the M phase portion of the cell cycle. Before we do that, do we remember any of the differences between sexual and asexual reproduction?
Asexual is where one organism makes a clone of itself, basically, and sexual is where there's two organisms going at it.
Thank you for that euphemism. Yes, you're right. So one organism in asexual reproduction is going to essentially make a clone of itself, a copy of itself. Sexual reproduction typically involves two individuals, one individual that provides sperm, a second individual that provides an egg cell, and from that is a process of fertilization. Josue reminds us that sexual is our origin stories. That's true. All living human beings are the result of sexual reproduction. Now, we've got to be careful, because what we're not talking about, and I trust that you all are mature enough to have this conversation, but we're not talking about sexual intercourse here. We're talking about sexual reproduction, which is simply the process of a sperm cell fertilizing an egg cell. And from that, you get a zygote, something called a zygote. And that zygote becomes an embryo, that embryo becomes a fetus, and that fetus will eventually become a more mature organism. But we can also think about this outside of the context of humans, and even outside of the context of animals. Plants do sexual reproduction as well. Fungi reproduce sexually. So it's just a matter of one organism that provides a sperm cell, the other that provides an egg cell, and those two cells meet. Does anybody remember the word that we call sperm and egg cells? It starts with a G.
Gametes.
Gametes, wonderful. And then everything else is known as a somatic cell. So let's do this. Quick joke break. This comes from Miss Fields. What did the cell say when his sister stepped on his foot? Mitosis. Okay. No laughs. That's all right. We'll move on. Mitosis. Mitosis is the cell division process used specifically for somatic cells. So sperm and eggs are gametes. Mitosis is not used to produce gametes. Mitosis is only used to produce other somatic cells, or it can also be used for unicellular organisms to reproduce. So this would be considered asexual reproduction. We talked a little bit about this yesterday, but I cannot underemphasize what's written in pink at the top of the screen. Interphase has to occur before mitosis. We have to copy the DNA. We have to replicate the DNA. The reasons for that will become pretty clear towards the end of the lesson today. Okay. So like I said, mitosis can be used to produce somatic cells, but it's also used for the asexual reproduction of unicellular organisms. Cells that are made up, or organisms that are made up of only one cell. This is how they make copies of themselves. Okay. Now, mitosis is one round of cell division. That will be in contrast to another process that we'll talk about tomorrow called meiosis. But mitosis is only one round of cell division. And the results of mitosis are two identical diploid daughter cells. There's a lot. That was a mouthful, right? So two cells that are identical to each other, and they're also identical to their parent cell. And those two cells, those new daughter cells, are also what we called diploid. This means that they have a full set of paired chromosomes. We typically notate this word diploid with this symbol 2N. Di, of course, means two. So what we're saying is they're twice the number of chromosomes. You get one from your father, one from your mother, and they're paired up. They are homologous chromosomes. Now, if we take a look at this image in the bottom right portion of your screen, we start off with what is referred to here as a mother cell. Often we'll just call it a parent cell. That's what I more typically will refer to it as. We see that the parent cell in this case has two chromatids. DNA replication has to happen first. So we double the amount. We double each chromatid. That's super important because if you don't double before you split, then each of the resulting daughter cells will have half the number of chromosomes. That's not going to work. So we've got to first double, then split. That's why DNA replication is so important. And in the middle, we can see the cell division process taking place. Mitosis has 4 steps, 4 phases. And we've got to remember the order of these phases. But they've got some pretty significant clues that make it a little easier for us to remember them. The first step is called prophase. The second step is called metaphase. The third step is anaphase. And the final and fourth step is called telophase. PMAT. The first step, prophase. Pro is a prefix that we've seen before. We saw it in the word prokaryote, and it means the same thing. It means before. In prokaryote, what we meant was it came before, a cell that comes before the nucleus. Here, we use the word prophase to mean before the other steps. Before the nucleus can actually be divided, these are the steps, these are the things that need to happen. Number one, the nuclear membrane needs to dissolve. So, of course, most or all eukaryotic cells have a nucleus, and that's the site of the genetic information. The nucleus provides protection for the chromosomes and the chromatin on the inside. So in order to have access to those chromosomes, we first have to get rid of the nuclear membrane. We've got to expose the chromosomes on the inside. So the nuclear membrane dissolves. This will often be represented in pictures with a dashed line, as you can see in the bottom right. You can see that the nuclear membrane here is starting to dissolve. Additionally, we talked about this yesterday, but DNA exists in the nucleus most of the time in the form of chromatin. And chromatin is like a big bowl of spaghetti. The DNA is totally randomly wound up within itself. It's very tangled. There's no order to it. That's not helpful to us when we're actually trying to make a copy of that DNA in order to divide it. So what it needs to do is organize itself, condense itself into chromosomes. And chromosomes, as we remember from yesterday, look like X's typically. We can also see them in the bottom right. So here's this process. The chromatin condenses into chromosomes. Lastly, before we can actually divide the DNA, centrioles and spindle fibers have to form. I like the analogy of a fishing rod. Some of you may never have been fishing. I didn't go fishing until I was 21 for the first time. So it's not the most common experience if you live in a city in a more urban environment. But I'm sure I trust that we all know what a fishing rod looks like and how it works. You've got to cast out that line. You let the line kind of wade back and forth in the water in hopes that a fish will think that whatever bait you've put on the line is actually a smaller organism that it can eat. Once you get a fish on the line, you reel it back in. I got the line stuck. Yeah, that happens too if you cast it back too wildly. But once that fish bites down on the line, you reel it in. That's a similar process to what we have going on here. The spindle fibers are like the fishing line, and the centrioles are like the actual reel. So the way that the centrioles work is that they're kind of like motors. They pull the spindle fibers in, and the spindle fibers attach to the chromosomes. That's what we will see in a couple of slides. They attach to the chromosomes. And so when the centrioles start reeling them back in, they pull the chromosomes apart. So in prophase, we need these centrioles and spindle fibers to form. After prophase is metaphase. Meta is a prefix that means middle. And here we can see that the chromosomes actually line themselves up in the middle of the cell. They line themselves up at the cell's equator, or I guess you could also say the meridian. And here you can actually see the spindle fibers attaching to the middle of the chromosomes, to the region of the chromosome that we call the centromere. That was one of our vocab words from yesterday. Spindle fibers are like the fishing line. They attach to the chromosome in its middle section, called the centromere. All right. After metaphase, we've got anaphase. Ana is a word that means apart, specifically to pull apart. So those spindle fibers that attach to the centromeres of the chromosomes, they rip the chromosomes apart. And so now instead of the chromosome being X's, they're just lines, they're sister chromatids being pulled apart from one another. And the thing that's actually doing the pulling is the centriole. It's reeling that spindle fiber back in. And on the other end of that spindle fiber is a sister chromatid. And then the last step of mitosis is called telophase. During telophase, the cells are being pulled towards the ends of the new cells that are forming. So telo is a prefix that means end. It also happens to be the end of mitosis, so it's easier to remember when telophase happens. But really, it represents the fact that the sister chromatids have now been pulled to the ends of the cell. Here we will also see two things. We will see the nuclear membranes starting to reappear. This time we've got two nuclear membranes because we're starting to create two cells. Secondly, we will also see the cell begin to cleave, which means it's going to be pinched in the middle because it's starting to separate into two new cells. So the cell membrane is actually being pinched in the middle. Okay. All right, and then after mitosis, we talked about this yesterday, but comes cytokinesis. Cyto means cell, kinesis means movement. Put it together, cytokinesis is cell movement. This comes after mitosis. It is not a step of mitosis. This is when the two cells officially split. All right. So I think this is a great image. And again, I'm a big fan of graphic organizers of pictures. They just give your mind something to grasp onto. And you can refer back to them as you're studying and taking a test. You have this visual image that you can think of. So I like this one. And you might think of drawing something similar in your own notebook, just to give yourself an idea of the fact that this is a cycle. Cells go through this cycle throughout their lifetime. And also it shows you the order of things as well. So I'll leave this if you guys want to take a screenshot or take a picture. Okay. So a quick practice problem. Let's see if we can place these pictures in the correct order. Which picture here do you think comes first? Now I will tell you, we start with interphase. So that's why there are 5 pictures. There are only 4 steps of mitosis. So one of these pictures is actually interphase. Which one do you think represents interphase and therefore comes first? Good. Thank you, Carolyn. Yeah, this is perfect. Number 2 is indeed the first picture, and thank you to Melissa. We can see that the nuclear membrane is still intact. We can see that there are organelles and ribosomes kind of strewn throughout the cell, so the cell hasn't begun any process of division here. Excellent. What comes next? What's the second picture? Excellent. Number 4, we can tell because the nuclear membrane is starting to disappear. It's a dotted or a dashed line. We can see that the centrioles have now appeared and the spindle fibers are starting to form as well. What about after that? Good, Josue. Good, Carolyn. Number 3, metaphase, because the chromosomes are literally lined up in the middle of the cell. That one's pretty easy to see. What about after that? Josue, Melissa, Carolyn, good job. Number 1, we can see anaphase as the sister chromatids are pulled apart from one another and they're moving towards the ends of the cell. And then that leaves us with number 5 as telophase. And we can see that the nuclear membrane is starting to reappear around the sister chromatids and the cell is starting to cleave in the middle. Good job. So you all will be asked to classify similar pictures like this on your test. So I'm glad that we can start to identify what steps of mitosis look like. One more concept for today, and then you all will have time to work on your asynchronous assignments. The reason we have to copy our DNA, the reason we have to have a synthesis step during interphase is because when we split, we still want the daughter cells to have the full set of chromosomes. If the parent cell starts off with 46, we need the daughter cells to also have 46. The only way to do that is to first copy or make a duplicate of the DNA. So we need to synthesize new DNA. So if we start off, in the case of humans, with 46 chromosomes, during the synthesis step of interphase, we copy that and it becomes twice the number, 92 chromosomes. And then when we actually go through mitosis and the cell divides, we have the correct number, 46. So in the case of a banana has 14 chromosomes. So what number should go in this middle cell here? 28. Thank you, Abigail. Yeah. And then in these two cells, what number should we have? 14. Good. So we start off with 14 in the parent cell. We have to end up with 14 in the daughter cells. And so to do that, we first have to double the number of chromosomes. Pretty simple. Okay, one more practice problem. What would be the results if mitosis or meiosis did not occur within the cell cycle? A, the genetic material would not be replicated. B, the cell would not grow large enough to support all of the new duplicated contents. C, the duplicated genetic material would not divide to make new nuclei. D, the cytoplasm or cell membrane would not separate, and it would remain all one cell. So if we take out mitosis, what is the result? What do you guys think? Okay, I've got people saying, C, the duplicated genetic material would not divide to make nuclei. That's correct. C is the correct answer. Now, let's break this down a little bit more and try to figure out when these other things happen. So when is the genetic material replicated? When does that happen? Interphase. Thank you, Janelle. Specifically, what part of interphase? Yes, thank you, Abigail. Yeah, it specifically happens during synthesis. So removing mitosis doesn't actually prevent the genetic material from being replicated. That happens earlier in the process. Okay, what about part B, question choice or answer choice B? The cell would not grow large enough to support all of the new duplicated contents. Which part of the cell cycle does that sound like? The cell not growing large enough. Which part of the cell cycle does that sound like?
Is it interphase?
Yeah, specifically which part of interphase, Lisa? Thank you. When do we see cell growth happening?
G1 or G2?
Yeah, in G1 and G2. So again, if something were wrong with the cell cycle process, and we couldn't do mitosis, that does not prevent the cell from growing because cell growth happens during G1 and G2 of interphase. And then answer choice D, the cytoplasm or cell membrane would not separate and it would remain all one cell. When does that happen during the cell cycle? When does the cytoplasm or cell membrane split? What's the step of the cell cycle when the cell membrane and the cytoplasm split up? What's that called?
Is it the M phase or mitosis?
No, mitosis is actually step C. So we were correct in saying that C was the right answer. Melissa just jumped in and gave us the right answer, but thank you, Lisa, for bringing that up. So mitosis is cell division. Specifically, it's the division of the nucleus. But when we're talking about the actual cytoplasm or the actual cell membrane splitting up, that's cytokinesis. That's when one cell becomes two new cells. All right, so answer A, that part was synthesis. Answer B is G1 and G2. Answer C is mitosis. And then answer D is cytokinesis. So we were looking for answer C. All right, so we've got 25 minutes, 26 minutes left in class. There is an exit ticket, Unit 3, Day 2 exit ticket. Again, guys, those warm-ups and exit tickets are important, even though they're not worth many points. They're important because, excuse me, they give you an opportunity to see what the test questions will look like. So please do not skip over them. Please make sure you're doing your warm-ups and your exit tickets every day during the designated time. We do also have an asynchronous assignment today. It's called Mitosis Practice. It should be a pretty quick one. The people in my last class were able to get it done in the given time, and I think we had even less time than you guys do. So go ahead and navigate back to Canvas and knock these two assignments out. I will be here on mute if you have questions. Okay, folks, it's now 1:20, so I appreciate everyone being here today and working diligently. I could look and see that most of you were getting your work done. For those of you who did not use your time wisely, we really need to rethink what we're doing. These minutes are built into class specifically so that you can get your asynchronous work done. It's not for you to go watch whatever or play whatever or talk to whoever or scroll on whatever. It's for you to get this work done so that it doesn't build up and you end up having to use your weekends to do it. So keep that in mind going forward. Let's be wise about how we use our time, and I will talk to you all tomorrow.
Is the mitosis practice formal or informal?
It is informal.
Okay.
See you tomorrow.
See ya. Have a good day, folks. Peace out.